The epithelial cells have certain specific and properly formed shape. For example, they may be cuboidal shaped, cuboidal cells, or they may be tall columnar cells. Compare this with mesenchymal cells which do not have a specific shape. They are just irregularly shaped like this. They do not have specific shape. So epithelial cells have a particular shape. Now epithelial cells, the second property of epithelial cells is that they are tightly adherent with their neighboring cells. They are tightly attached with their neighboring cells through certain junctions. Basically, we will not go into those details, but basically they are attached with the neighbors. Again, the mesenchymal cells, they are just randomly interspersed in typically in a connective tissue. So they are not tightly adherent with their neighboring cells. Now here you can see that these are the tall columnar epithelial cells and they are tightly adherent to their neighbors like this. And here you can see that there is just a single layer. You can see there is just a single layer in this plane. There is just a single layer of these cells. So this type of epithelium is simple columnar epithelium. And by the way, if you are wondering why I am teaching epithelium, you might be thinking that you have clicked a wrong video. No, it's not wrong video. It is actually about the neuroembryology and it is actually about the histogenesis of neural tube. But with this basic concept, it will become easier for you to understand the histogenesis of neural tube. So that is why I first of all started discussing the epithelium and then step by step we will go into the details. So this is the simple columnar epithelium. It is single strata or single layer. Strata mean layer. So it is single layered and that is why we simply call it simple columnar. If it was cuboidally shaped, then it would be simple cuboidal epithelium. Now if we move forward, then of course let's suppose here is another type of epithelium, and in this case you can see that this is not columnar but it is cuboidal. And the second thing is that you can immediately see that we have two layers here. So this is the basement layer, the basal layer, and then we have this other layers. Let's suppose here is the basement membrane. So we have two layers. So and we have, you can see, properly marked boundaries of the cell, and that's why we have two layers. And that is basically stratified. If there are more than one layer, it is stratified. It may be 3, 4, 5 and so on. It may be many layered. Basically strata mean layered, layer. So stratified, and of course depending upon the shape, in this case it is cuboidal epithelium, stratified cuboidal epithelium. It may be stratified squamous epithelium and so on. Now let's move forward. That here there is an interesting scenario. Now here you can see that if you try to focus, you might be tempted to label this one as stratified epithelium. You see, this looks like here are these nuclei of the cell. So they look like that this is one layer of nuclei, and then there is another layer of nuclei. So they look like that this is stratified squamous, not squamous, basically columnar. So it looks like that it is stratified columnar, right? But it is actually not stratified. Let me show you and let me explain to you why it is not. I think in this diagram it would be difficult for you to understand, so let me basically draw a sketch here for you. So here let's draw a cell. So if a cell is like this and then there is neighboring cell like this, in this way, so basically this is a single layer, right? And you see that from top to bottom its cell membrane, it's touching the top here and it is touching the bottom here. Now if you see that if there is, let's suppose, a limited space and the cells have to proliferate within that limited space, and there is not much cytoplasm inside the cell, then what will happen? That let's suppose here there will be this cell like this, it will become like this, and here there will be nucleus, and then here another cell, neighboring cell will come, and it will sort of form like this, and its nucleus will be somewhat in a upper position, right? And then there may be another cell here, right? And again its nucleus might be in a lower position, and here its nucleus might be in a upper position, and so on. You can continue with this. So now basically if you look carefully, the cell membrane, it is touching the top and bottom of all the cells. This is first cell, its cell membrane also touching the cellular process or cellular membrane, basically touching here at the top, this one also touching, and bottom as well. So basically they are lengthwise, they extend from top to bottom. The cells, they extend from top to bottom lengthwise, they are totally extending from top to bottom, but basically the position of their nuclei are different, and that is why due to different position of their nuclei, when you look especially with a low magnification microscope, they look like that there are multiple layers of the cells, but actually they are not multiple layer. This is single cell and they are touching from top to bottom. This is other cell again touching, but here this part is narrow. So if I draw again here, it should be, let's suppose, like this. It should be like this, but what happened that this one somewhat narrowed out because of lack of space, perhaps. Actually there are numerous other reasons. There are some molecular reasons which are not precisely known, and of course I do not want to go into that debate as well at this time. It's a bit complicated. So basically if they are growing in a limited space, they are sort of jumbled up like this. So one nuclei sort of shift here, one nucleus sort of shift here, the other nucleus sort of shift here. Wherever they get the space, they sort of jumble up together to fit in a limited space, right? So this type of epithelium, which especially on low magnification look like it is stratified, but it is not actually stratified. It is simple columnar epithelium, but it looks like stratified. It is false, it is pseudo, so it is false stratified epithelium and columnar epithelium actually. So we call it pseudo mean false, pseudo-stratified columnar epithelium. So this is unique type of situation. Again, I am telling you that if you are viewing at a low magnification, and you can easily, typically on under the microscope, you can easily view the nuclei, and if you are looking just at the nuclei, you may be tempted to think, okay, this is one layer, this is the other layer, so this is simply the stratified epithelium, but it is actually not. Again, if you focus here, if you focus on this 3D model as well, you can see that the cells are arranged in a unique fashion. They are jumbled up together in a limited space, so it looks pseudo, actually it looks falsely stratified. So that is pseudo-stratified epithelium. And as I've told you, that cells are proliferating in a limited space. So here if you focus, that with the property of the pseudo-stratified epithelium is that more cells can jumble up in a limited space. So for example, if you count, these are of course almost the similar sized epithelium. So here if you count 1 2 3 4 5 6 7 8 9 10 cells are there, but here if you count 1 2 3 4 5 and so on, I do not want to bore you with all this counting, but I hope you got the point now. So what happens now? Let's come to neural tube. That yeah, we have our beloved neural tube. Where it is? So here we have our beloved neural tube. Now basically, actually, initially the neural tube is simple columnar epithelium. Neural tube is initially made up of simple columnar epithelium. Now this neuroepithelium, basically the neuroectoderm, initially it's the neuroectoderm. It is single layered simple columnar epithelium. But as these cells proliferate within this limited space, later we will see that actually meninges are there, pia mater is there, and then here there are the junctions are formed. There is not much space. That cells are typically not allowed to come here and form another layer, so they have to grow within this limited space. So what will happen, that again they will jumble up together. As they proliferate, they will sort of jumble up together, and this simple columnar epithelium, it will be converted into this pseudo-stratified columnar epithelium. And the other thing is that here you can see that there are these cellular processes. Here you can see there are these cellular processes, and these cellular processes at the basal side, here is the basal side, and here is the luminal side or apical side. So these cellular processes, they meet with their neighbors. Basically they are also called end feet or end foot processes. They meet with their neighbors to form this continuous membrane like structure here as well as here. These are called external and internal limiting membrane. Let me explain this concept to you. I know many of you might be confused, so don't worry, Dr. Aizaz is here to clear up your misconception. So basically here is, let's suppose, one cell, and here there is this dilated end, right? And here also there is a dilated end. Now there might be now another cell here, and like this, and they are also having these dilated ends like this. Now these dilated ends, they are lying close together, and there are certain proteins which are connecting. They are basically called junctional complexes. So junctional complexes are connecting them together at the top as well as bottom, and they are forming sort of a membrane like structure, and this membrane is called external, and this is external, this one is external from this one is at the outer side, this one is external, and then there is internal. So external as well as internal limiting membrane. And of course, please don't forget that at the site of external membrane, this is basically the basal side. Here we have the basement membrane, which consists of some collagen fibers, and basically connective tissue is there. So basement membrane along with these foot process, they are called external limiting membrane. And some literature also say that of course outside there will be the pia mater. So these three things, they are basically forming the external limiting membrane. Or some literature, they say two things are there, basement membrane and these joint foot process, but other also include the pia mater into this external limiting membrane. But I hope you got the concept. Similar foot processes are also present in retina of your eye, and there also they are forming the external and internal limiting membrane. There these limiting membrane, they are formed by Muller cells. Muller cells are special type of radial glial cells that we will discuss later. We have radial glial cells as well. So anyhow, let's move forward, and of course don't forget that we have lots of mesenchymal cells which will proliferate and which will form meninges around it. We have pia mater, arachnoid matter and dura mater, but here I have just shown the pia mater. Basically don't forget we have three meninges, three meningeal layers basically, but here Pia matter is relevant, that's why I have shown that. And from where this mesenchyme come for forming the pia matter, in literature it is said that two sources are important for the formation of meninges. One is neural crest cells, mesenchymal cells that arise from neural crest cells, and the other is that there is some contribution from the mesenchymal cells that are derived from paraxial mesoderm. So neural crest cells as well as some contribution from paraxial mesoderm, they will proliferate and they will form irregularly shaped cells, mesenchymal cells. As I've told you, they do not have particular shape, they are irregularly shaped and they are just randomly interspersed within the connective tissue, embryonic connective tissue, forming the mesenchyme. Now here I have isolated just a single cell, a single neuroepithelial cell that was forming the neural tube out of many lots of neuroepithelial cells that are forming initially the neural tube. I have just isolated a single cell so that it will become easier for you to understand. And here is the pia matter, a piece of pia matter here. And of course here there will be the lumen. This is the luminal side, which is also called ventricular side or ependymal side, because ependymal cells will form here, we will discuss later. And again here you can see these end foot, and they will of course meet with the neighboring one. And here this is the external end foot, here it will also meet with the neighbor external end foot, and again it will form the external limiting membrane here and internal limiting membrane at this site. Now what will happen that these cells, they will proliferate, they will increase their population, and ultimately almost all of them, they will transform into another type of cells. And this type of cells, they are called radial glial cells. They are called radial glial cells. This is important, and they are the hero of this show, radial glial cell, hero of the histogenesis of neural tube, because most of the cells, they develop from the radial glial cells. And glia in neuroglia, basically it is the part of neuroglia. Glia basically, actually glia means glue, but neuroglia typically refers to sporting cells of the nervous system. So in this context, they are not only sporting cells, but they are also the precursor of many different types of cells of our central nervous system that we will just discuss. Now what will happen that they will undergo, these radial glial cells, they will undergo two types of cell division. One is the typical type of cell division where this radial glial cell, it will proliferate, and let's suppose here is a single radial glial cell. It will proliferate and it will form two daughter cells which are very similar to this one. I mean, it will form similar cells as this one. It will form two radial glial cells. That is just like every other cell in our body. And this type of cell division is the typical cell division, which is also called symmetric cell division. Symmetrical cell division. Now what will happen then, some of these glial cells, what they will do is that they will then transform into, they will undergo such type of cell division that one of the daughter cell, it will remain similar to this one, its parent. I mean, they will form one radial glial cell, and the other cell, that will be very different from this. Right? So this is basically asymmetric cell division. Now this will go to another path. This is basically like a stem. So stem of a tree, that here it was going straight and then it took another path, and here it was going straight and took another path like this. So this is like a stem. So these are basically stem cells. By creating this type of cell, they are basically self-renewing themselves. For example, if both, if this was converted into two different types of cells, then this cell would have been lost, right? Now, because it has created a copy of itself, it has self-renewed itself basically, so it can now generate more of these type of cells. So it will again create a and similar cell and again a different type of cells. So you see stems are being formed here. So this is that's why these radial glial cells are also called stem cells. Stem cell is a whole lot of different topic, and it's a vast topic. I'm not going into detail of stem cells. Basically they are multipotent, they can form different types of cells. Here also they will form different types of cells. So anyhow, a basic concept is necessary. I know this topic is a bit complicated to understand, so I'm trying my best to simplify it for you, and I'm trying my best to explain all the related concepts that comes into the way, so that you develop a crystal clear, overall good crystal clear concept, not just about this topic but overall about the basics of histology and embryology and physiology and whatever related topics that come along the way. So anyhow, I think we wandered a lot, so I'm sorry for that. So anyhow, what we were saying, that this type of asymmetric cell division, and basically it is self-renewing itself. So that is another important property of stem cells. So these are basically stem cell as well as the supporting cells, and I will tell you why I am saying that these are supporting cells. So basically now what will happen, that they will undergo asymmetric cell division, as I have told you previously that it has underwent asymmetric cell division, and as a result of asymmetric cell division, this cell has not been lost, it has not been transformed into this cell. Basically what it did, as I have explained earlier, that basically it made a copy of itself, it self renewed itself, and then the other one is this cell. It has formed this cell. Now basically this cell is the precursor of neurons. So it will form a lot of neurons. It will blast the neural tube with neurons. By blast I mean it is not that type of blast. I mean with the blast or the population blast. There are lots of neurons formed as a result of these cells, and that is why they are called neuroblast cells. So basically the radial glial cell, radial glial cell, in this case the single cell, it has underwent self renewal, as well as it has formed a neuroblast cell, and it is apolar. It in this case, at this time, it does not have the processes. Later we will see that processes developed. Right now it is apolar neuroblast, right? So apolar neuroblast has been formed. Now what will happen, that this apolar neuroblast will become bipolar. How it will become bipolar, that it will form two axon shaped processes, right? Like this, two similar axon shaped processes, and then it will ride on this radial glial cell like a monkey or like a snake, like this, and it will reach to this top. Actually it's not top, towards the periphery, you can say, towards the away from the ventricular side, and basically this is the basal side. So towards the basal side or towards the external limiting membrane. It has written in many different ways in the textbooks. I want you to have a clear concept, so when you read any book of neuroembryology, you get the concept, because basics are same, but the way of writing is different. You don't need to cram the topic. No topic in medical or no topic in sciences should be crammed. Actually, everything should be properly understood, right? So it was this bipolar neuroblast. Initially it was apolar neuroblast. Now it is bipolar neuroblast, and this radial glial cell, it acted as a scaffold to provide the support and to help reach this neuroblast to its destination. and now one of the process will be lost and now it will become unipolar. Well, honestly speaking, I'm not sure that why initially it becomes bipolar and then this process is lost, but here is my own hypothesis. Bear in mind it may be totally wrong, but at least it will help you remember. So my hypothesis is that it initially develops these two similar process, axon shaped processes, and they help this cell to climb up like a monkey or snake. They help this cell to climb up. If it was unipolar initially or apolar initially, there might be difficulty in this climbing up process, right? So that is why initially, that might be the reason that initially the bipolar neuron forms, and because we only need single neuron, here there will be dendrites that will develop and this process is lost, right? So it has again become unipolar, and here ultimately dendrites will develop and axon will become longer and mature neuron will be formed. In this way, lots of radial glial cells will form a lot of neurons, mature neurons, right? So a lot of neurons are formed. Now here, if you look carefully, you can see that initially here was this single layer which contained these cell bodies of the radial glial cells, right? So this is near to the ventricular side or near to the ependymal cells will form here. So that is why this layer is called ventricular layer or ventricular zone or ependymal zone, ventricular zone. Then outside it, another layer is formed, and this layer consists of lots of cell bodies of and dendrites as well, cell bodies and dendrites of neurons. This is the main layer. This is most important layer. This is the crucial layer, and that is why it is called mental layer. It's not mental, it's not psychopath, it's mental. Mental mean it's important. So mental layer, the primary layer basically. So it's the mental zone. Oh, what I have written zonal, mental zone. And finally the axons are in the last layer here. And what is the name of this layer? This is the outermost layer. It is called marginal layer. Marginal layer or marginal zone. All these zones can be replaced by layer. So marginal zone, mental zone and ventricular zone, and because this mental zone is in the middle, it is also called intermediate zone. Intermediate zone. Right, so let's move forward. Now here it is also worth mentioning that the radial glial cells, they always extend from this internal limiting membrane up to the external limiting membrane, or you can say they always extend from the ventricular side up to this pial side. Pial mean pia matter, it is facing the pia matter. So basically they extend all the way throughout this neural tube. So if more and more cells are formed, so they may extend their process like this, and they may become longer like this, right? I hope you got the concept. Now when all the neurons that are required initially, when all the neurons are formed and further formation of neurons cease, then the radial glial cells, they worry about supporting these cells, supporting these neurons. Now they think about the needs of these neurons, their needs of particular ionic balance requirement, their support, their nutrition and so on. So what will happen that they will form some cells that will form macroglia of neurons. Macroglia of central nervous system, macroglial cells of central nervous system. The important one being the astrocytes. These star-shaped cells, these are called astrocytes, and basically they are in both zones, the ventricular as well as the marginal zone. and also along with that, the dendrocyte, oligodendrocytes will form. Oligodendrocytes, they are involved in myelination of neurons. We will discuss this process later. And the astrocytes, as I've told you, they have many important functions. We will not discuss in this lecture series. We might discuss when we will talk about neurophysiology or neurohistology. Then we will talk about the astrocytes. They are important for ionic balance, for the nutrition, for the maintenance of blood-brain barrier. These are very interesting topics that we will discuss someday later, not today. And of course, oligodendrocytes are here. So astrocytes as well as oligodendrocytes, these are two important glial cells that are here. Then what will happen that the radial glial cells, the hero of this show, they have created the neurons, they have created the macroglia, the glial tissue, the glial cells of the nervous system. Now they want to relax and they want to retire. So what they will do is that they will bring back their process and they will transform into another different type of cells, and these cells, they are lining the luminal surface of neural tube, and these cells are basically called ependymal cells. They are called what they are called ependymal cells. And that is why I said that this part is also called ependymal zone. This is ventricular zone or ependymal zone. This is the which zone? Mental zone, and this one is which consists of lots of axons of neurons. This is basically the which zone? Marginal zone. And here another important thing that I want to mention, that oligodendrocytes, they will myelinate these neurons, not neurons, these axons of these neurons, right? And as a result of myelination, myelin sheath consists of lots of fat, and fats are typically white colored. So this layer will be especially more of white colored. It will be more of white colored, and this will be sort of a bit darker due to presence of these nuclei of the cell bodies of neuron. So basically this is the gray matter that is formed here, gray matter, and this is white matter. So white matter. So marginal zone consists of white matter and mental zone consists of gray matter. And by the way, astrocytes, they are present in both of these layers, but the what are these oligodendrocyte, they are mainly present in the marginal zone. There is actually a mistake in this diagram that I haven't shown the astrocyte in the marginal zone, but they are actually there. While creating these 3D models, I forgot about this, but now as I am giving this lecture, now I remember that they are in both of these zones, and in this marginal zone they have specific functions. Basically, astrocytes are of mainly two types, protoplasmic and the other one I forgot its name. Yeah, they are, I think, fibrous astrocytes, if I'm not wrong. Anyhow, these things are about the histology. I do not want to go into those details. Oh yeah, I remember another important fact. You know, these radial glial cells, they are also the member of astrocytes family. They are basically cousins of astrocytes. So we can actually say that basically we have astroglia. We have astroglia family, and astroglia consist of astrocyte, and we have the radial glial cells, which are also type of astroglia, radial glial cells. And of course, astrocyte, we have two types, protoplasmic and the other one is, I think, fibrous. Anyhow, now let's move forward, and I have been calling these type of glia as macroglia. What about microglia? Actually, later in development, don't ask me when, because I do not remember the dates. I have very bad memory. Even my wife gets angry with me because I often forget her birthday. You can look any reference textbook to see when. But anyhow, later in the embryonic life, when blood vessels develop within the central nervous system, these blood vessels, they will bring special type of very small glial cells. These are Microglia. Here you can see microglia. So these are the microglial cells. Microglial cells are basically specialized macrophages of central nervous system, and they arise from the yolk sac. We will not go into detail, but they arise from the yolk sac, and they are basically antigen presenting cell. They are actually the part of immune system of your body. So central nervous system have specialized type of macrophages, and these are actually called microglia. So ultimately this is the situation of neural tube. So I hope you can understand, and if I show you here that by the way, ependymal cells, they are ciliated and they are simple columnar epithelium. They are single layered, they are not stratified, or they are not even pseudo-stratified. They are simple columnar, right? So we have this layer here. Let's use another color. We have this layer here, this layer, this whole one, actually this one. This is the ependymal zone or ventricular zone of neural tube. Ependymal or ventricular zone. Then we have this zone of gray matter, which consists of cell bodies of neuron, as well as the dendrites as well. So this is which zone? This is mental zone. This is mental. It's not crazy, it's important. So mental zone, right? And then we have these axons, right? So we have these axonal process. This is the forming the white matter, and this is marginal zone. This is the marginal zone. And of course we also have these microglia, these red one microglia interspersed in between. We have these astrocytes, we have oligodendrocytes, and so on. And this mental zone is also called intermediate zone. So ultimately this is the situation of neural tube. I hope you can understand everything. Please look at this 3D model carefully, it's very important. It is basically a cut section of neural tube. At this stage we have, let's suppose, cut a part of neural tube, and this is the luminal side, this is the pial side or marginal side. So do you really understand it? I hope so that you understand. You should actually understand, but anyhow, if you do not hesitate to write the comments below, and you can ask the questions freely. No question is stupid. Even if you think it's stupid, it is not. We all are once beginners. So if you do not understand something, do not be ashamed. By the way, it's not possible for me these days to answer each and every comment, but I try my best to answer as much as possible. I'm really sorry, sometimes I'm not able to write the comment, reply to comment, but I try my best to do so. So finally, let's review all these things on a PowerPoint slide, animated PowerPoint slide. Bear with me, it won't be the typical boring PowerPoint slides that you see in your medical college. It would be much more interesting. I'm not being a narcissist. I have worked really hard on this lecture, so I deserve a little self-praise. Allow me to do so. So here we have these neuroepithelial cells, and they will be transformed into radial glial cells, which are hero of our show. Then these radial glial cells, they will form apolar neuroblast, and then they will convert into bipolar neuroblast, which will climb on these processes of radial glial cells like a monkey or snake, and ultimately they will reach into their destination, and then they will lose one of their process, and then they will form lots of dendrites here to form the mature neuron. That was about the formation of neurons. When this process is completed, only then these radial glial cells shift to formation of glial cells, right? So they will transform into glioblast. I deliberately skipped these details when we were talking on the 3D model. So basically these are the glioblast, or they are also called spongioblast. They are precursors of astrocytes and oligodendrocyte, and glioblast they will convert into astroblast as well as oligodendroblast, and of course ultimately they will form astrocyte as well as oligodendrocytes. Typically these processes, both of these processes, I mean the formation of astrocyte and oligodendrocyte, they are going on side by side. But for the sake of explanation, I first talked about formation of astrocyte and then I talked about formation of oligodendrocytes. And finally, the radial glial cells ultimately they will transform into ependymal cells. Here, honestly speaking, I'm not sure that how it actually happens, whether all these radial glial cells they themselves transform into ependymal cell, or there it is possible that they divide into ependymal cell and they themselves they are lost, right? So to be honest, I am not really sure because I could not find a proper reference in any of the literature that I have studied for this lecture. So if you know a proper answer, please let me know in the comment section as well. And we have this yolk sac, yellow one. So as the blood vessels they develop within the nervous system, what will happen there, they will bring the microglial cells into the nervous system. So as you can see that from this diagram, it is clearly evident that most of the cells actually of central nervous system, they develop from the radial glial cells, except the microglial cells, which are basically specialized macrophages of central nervous system. These are the immune cells of central nervous system. And by the way, these astrocytes, oligodendrocytes, and ependymal cells, they are the part of macroglia. And these microglial cells, as their name is microglial, so these are basically microglia. They are the part of microglia. Of course, don't forget that radial glial cells, they are actually the part of family of astroglia. So radial glial cells are also the part of macroglia. So 1, 2, 3, 4, all these 4 cells, they are the part of macroglia, and this one is the part of microglia. So this was all about the histogenesis of neural tube. I hope you learned something from this video. Thank you so much for watching this video. Have a great day. Goodbye.